welcome to this part where we're going to focus on some of the basic methods and principles in cognitive psychology. So we'll start by thinking about some of the research methods that cognitive psychologists can use to study these inner mental processes. And there's three basic approaches. So one of these is we could use lab-based, so laboratory or experimental studies. We can use psychobiology, so this is looking at the internal physiological responses, so whether that's studying changes in neuronal response in the brain or measuring peripheral responses such as the amount of skin conductance or sweat that somebody has given off that can indicate internal processes. And then finally we're going to focus a little bit on how we can use individual case studies, and particularly this would be patients that might have had damage to a particular region of the brain, and how we can use this information to inform normal, healthy functioning. So start off, let's have a little think about what we can use laboratory or experimental designs. And throughout this course we're going to cover many different examples of cognitive experiments that are done on a computer to examine the way that you process stimuli in different ways. So typically you might design a carefully controlled experiment that would use a computer program, it's a piece of software, and you ask participants to perform tasks in a controlled environment. So typically you would invite them to come into the lab and sit down and do the study. During contemporary times, a lot of this stuff has actually moved to online. So even though we say laboratory, I think what I'm probably referring to here is more just careful computer-based experimental designs. Now the real strength of taking this approach is the fact that you've designed everything is that you have really careful control over all of the stimuli, so what people are viewing and experiencing, and you can vary one element that you're interested in and see how that influences people's reaction time or their accuracy on the task. So again, the benefits of this is that you've got relatively standardized ways that you can design and then analyze this data so that every participant is experiencing a similar set of stimuli, and it gives you a high probability of drawing valid inferences. So if there's only one thing that you're manipulating or varying, and you end up seeing a difference in the way that people respond to different trials, you can be pretty confident that the thing that you're manipulating is the thing that is causing these differences in the reaction time or the accuracy rate that people show on these experimental tasks. Okay, one of the difficulties or challenges with using the experimental approach to understand human behaviour is that sometimes you could argue that experimental approaches lack ecological validity. So in other words, the findings and the results from experimental studies are not necessarily always applicable to real life and everyday behaviour and cognition. And one of the things we'll go as we go through this course is you'll have the opportunity to take part in lots of cognitive psychology experiments. And one of the things you'll notice is that a lot of the tasks and the stimuli that you're presented with can be a little bit artificial. So you're being asked to do things you wouldn't necessarily encounter in everyday life. Now, that does sometimes present a problem. However, I think on balance, generally the experimental approach is fundamental and it's been really important for understanding the basic cognitive processes that occur. But it's always worth paying attention to what is the task that's being done in an experiment, and what are the researchers claiming that performance on this task relates to. And it's important to always pick up with how closely do these two things align? How closely do the sort of methodology align to these theoretical constructs that someone is talking about? So as an example of a cognitive psychology experiment, what I've presented on the screen here is a illustration of an experiment that I ran a few years ago. It's written using open source software called PsychoPy. And in this experiment, I've used something that's known as a Simon task. So in the Simon task here, what you end up having is stimuli, and these stimuli are coloured circles, are presented on the screen and are associated with the correct response option. So for example, participants would see a blue circle that would appear on either the left or the right hand side of the screen, and every time that you see this blue circle, you must press a corresponding button using either your left or the right hand. Now in this case in the experiment we set it up so the left index finger would be always over the blue response key on the keyboard, and the right index finger is hovering over the yellow coloured key on the keyboard. So it's a pretty simple task that you've got here. Every time you see a blue circle, press your left finger. Every time you see a yellow circle, press your right finger. Crucially though, the responses end up being driven by an irrelevant stimulus dimension, which in this case is the location at which the circle is presented. So if you've got the center of the screen in the middle, these yellow and blue circles, so there only appear one on each trial, but they could appear either on the left or the right hand side of the screen. And for trials that are compatible, you would end up having the coloured circle appear on the same side as the response key. So in this case, if our left index finger is over the blue key and our right index finger is over the yellow key, when the blue circle appears on this side of the screen, on the left hand side, we're faster to respond to it than if the blue circle appears on the right hand side. Now when I say faster, we're looking at a difference here of sort of 20, 30, 40 milliseconds. So it's small differences. So if we remember that the location of the target appearing, that's a dimension that for us is irrelevant. So if we see a blue circle, it shouldn't matter to us whether it's appearing on the left or the right hand side of the screen. Our only job is just to press that correct button with our left index finger. Now the problem is, when the circle, so if that blue circle appears on the left hand side, it primes a little bit for us to be more ready to make a response with our left finger. Similarly, when it appears on the right hand side of the screen, we're a little bit more primed to make a response with our right finger. And we've got to kind of resolve this conflict that we have and make the appropriate response here. So let's see an example. Whenever I explain these, it always seems like very theoretical and might be hard to understand. So if we see an example, you can actually maybe get a little bit of a better flavour of what is actually going on, and which trials you find easy and which you find a bit more challenging. So I'm going to give you a few circles that appear on the screen coming up here. Every time that you see a blue circle, I want you to tap your left finger as quickly as possible. So imagine now that this left finger is blue. You activate this every time you see a blue circle, and the right hand finger, every time you see a yellow circle, you tap this. And in the experiment, you'd want to be doing this as quickly and as accurately as possible. So we tell participants we don't want to sacrifice accuracy and be guessing, but to try and move as quickly as possible, making sure that you get the correct response. Okay, are you ready? Let's try a few of these. So we've got a fixation cross that appears in the middle, and then after a delay of around about 750 or 1 second, you'll see the colour appear. So there we go, we should have pressed our left index finger. Let's have a look at the next one. We've got the fixation cross and then your right finger. These are two examples of compatible congruent trials. So they're appearing on the same side as where you would want to press your finger. So you should find these pretty easy. You should be fastest and most accurate responding to these congruent trials. Next one. Here is where we have a bit of a conflict. The correct response was with the right finger, but it was appearing on the left-hand side. Okay, so one thing you should notice in there, that we are much quicker to respond to those congruent trials, and it's a little bit harder when you've got this mismatch between the location and the response finger that you're meant to be using. So in this experiment, what I was interested in is studying how cognitive control is affected by the amount of incentive that was up for grabs on each trial. And this is beyond the scope of what I expect you guys to understand or think about right now. But the specific kind of theory or the rationalization for this experiment is the idea that when you're promised a incentive, this can lead to a very temporary sort of spike in dopamine levels in the midbrain. And that's suggested to interfere with our ability for cognitive control. So what we did in this study was experimentally manipulate on each trial the incentive that was there, and we varied the magnitude of this. So there were low, moderate or high incentives. So some trials were worth 1, others were worth 5, and successful performance on the high incentive trials was worth 40 baht for getting that correct. So I think a relatively substantial reinforcement or incentive for performing well. We also had some mixture of the valence. So some trials, if you perform correctly, you would gain money. On other trials, you had to perform correctly to avoid losing money. And what you end up seeing illustrated in the figure here is very typical of cognitive psychology experiments that describe each trial in terms of a time series. So you start off kind of moving from the top left to the bottom right, with each screen is representing what is being presented on the screen to participants. You would normally see the amount of time it's presented. So in this experiment, for 1000 milliseconds, so 1 second, we present a fixation cross. You want to present a fixation cross to draw participants' attention to the center of the screen. And again, this is going to be common throughout most experiments that we do. We don't want participants' attention to be wandering around to different locations. We want their attention central. And it's quite important, especially in this experiment. Then we present this incentive for 750 milliseconds, so it's long enough for them to consciously process it and be aware that, yes, this is what is on offer for the current trial. We then put a blank screen, so there's a variable delay of between 0 and 200 milliseconds before the stimuli, so the blue or the yellow circle, appears. One of the reasons that we put this in is so that people can't, or participants can't, preempt and guess when the stimuli is going to appear and make a response as quickly as possible. We want them to be actually reacting to the appearance of the stimuli. Then the stimuli appears on the screen. So in this case, a yellow circle is appearing on the left-hand side of the screen. The participant makes their response on the keyboard, and after 1.5 seconds, we give them some feedback. So in this case, they've pressed the correct response, which is the right keypad, and we tell them, well done, this is how much you've earned for that trial. So what we would then examine afterwards is their average reaction time and error rates on all of the different trial types. So depending on the magnitude of the reward that we were offering, also the valence here, how does their average reaction time in these trials and accuracy differ? Now have a think, how many trials do you think I would probably ask people to do? What number do you think would be reasonable? So it might shock you the answer of this, but in many cognitive psychology experiments, it's a lot because the differences in reaction time between different trial conditions can often be quite small. So as I mentioned earlier, we can aim for differences that are low as sort of 20 or 30 milliseconds. So we want as many trials as possible to be confident that if we end up finding differences in reaction times across different conditions, that these are not down to just kind of random fluctuations in trial by trial performance, but that they're actually reflecting genuine effects or genuine differences across the conditions. So to do this, the more trials that we can get somebody to perform, the more confident we can be in their average reaction time. So in this study, what I ended up giving people was 288 trials. So this would take up to around about sort of 40 minutes to do this. So again, the benefit of this is that we can be relatively confident that any differences in reaction time and error rates across the different trials is caused by the manipulation of trial incentives here, and not by anything else, as every trial that people are doing is essentially the same apart from the incentives. So that is the only thing that's changing across these different trials. Now what I want you to do is just have a think, in this study, what might be some problems that we encounter that mean our data isn't as valid as we would like it to be. So pause the video and just have 10-15 seconds to think about a response to this. So one pretty reasonable possibility is that people just start getting bored during the experiment. So they stop paying attention to each of the trials. Maybe they start guessing on their responses. They stop focusing on the incentives. So we then need to be very careful in the way that we analyze this data. So for example, if people don't pay attention to the incentives, one way we could check this is that we could ask people on certain catch trials. So after they've done 50 trials, we ask them what was the incentive that you were promised on the previous trial. And if they're consistently not able to tell you, maybe that's a pretty good indicator that that's somebody that wasn't paying attention to these incentives. And again, if you've got someone that's not paying attention to the incentives, then they're not paying attention to the thing that you're manipulating, and their data doesn't mean much. It might also be the case that with this boredom, people just start guessing their responses rather than waiting until the stimuli appear and properly responding or reacting after its appearance. So instead, you're thinking that whatever happens, I'm just going to press the left key button response. Again, the way that you can check this afterwards is you go through that data and you might look at the reaction times. If somebody is too quick, that would be a red herring. So if they respond faster than 200 milliseconds, it is likely that they're guessing on these trials rather than reacting to them, because we know that the threshold of people making responses is around about sort of 150 milliseconds. You might also then start to look at whether there's a trend in their accuracy and reaction time. So if they started off being very accurate and then towards the end of the experiment they're only with a 50% accuracy rate, that's another good indicator that somebody is guessing on these trials. Finally, what can we say about the ability to generalize findings from this outside of the laboratory? What challenges do you think exist? So this is one study that that examines this idea of cognitive control, and I think could be linked to explaining why individuals choke under pressure. So if you look in the sporting literature, you've got lots of instances and examples of athletes and normal people that when you're in a high incentive performance domain, so you've got the opportunity to win lots of prize money or get lots of prestige for executing a skill such as putting a golf ball. In those instances where your performance can lead to a really massive payoff, athletes very often choke, so they perform suboptimally. And there's some way that you could kind of link the findings from this as a partial explanation for choking under pressure. But I think we've also really got to constrain and realize that the stuff that people are doing here, responding with their left and the right index finger to very simple stimuli, is incredibly different to the sort of same real-life choking that occurs in sporting domains. So that was a bit of a kind of complex dive into an example of an experiment that you might run. One principle that is important with these lab studies, and particularly with using reaction time data, is that we use this concept of mental chronometry. So we try and use reaction times to make inferences about the cognitive processes that are going on. So if we take a much more simple experimental design, and imagine that you have an experiment and all we want to do is work out how long it takes people to make a simple decision of whether to press the left or the right key. So in this case, this is a version of Donder's reaction time experiment, and we've got two different versions in here that we give people a simple reaction time task where they see a light appear on the screen, and as soon as you see the light, you've got to press a key button response. And if we get people to do that, your typical reaction time on this might be 150 milliseconds. Now, if we give people a choice reaction time task, and in this case we're presenting stimuli that might appear on the left or the right hand side of the screen, and similar to the Simon task, you've got to choose whether you respond with your left or your right finger to different key button responses, then we can look at the reaction time that people take to make appropriate responses for this. And obviously what would typically happen in these experiments is that participants would take longer to respond in the second type of experiment where you've got to make the decision. And then you can compare the difference in these reaction times so in one experiment you are simply engaging in perceptual processes of seeing the stimulus on the screen and then executing the motor response. In experiment 2, we've got perceptual processes, but then we've got some decision making, and then we have the same executing the key button response. And if we look at the difference in those reaction times, whatever difference there is can be attributed to cognitive processing. So we can see how long does that decision making process take through this method of subtraction. So we've just seen in the previous section how cognitive psychologists can use experimental tasks and analyze the reaction time and accuracy data from those tasks to make inferences about the cognitive processes that are occurring during that task. Another approach is to use cognitive neuroscience and psychophysiological measures to try and provide some information about the brain activity whilst people perform cognitive tasks. So psychophysiology encompasses the examination of animal and human brains using psychobiological measures. So some common examples of these are using electrodermal activation. So this is where you would attach some small electrodes, typically to the fingertips of a participant, and you would measure the level of skin conductance. So as the sympathetic nervous system is activated, this leads to a very small increase in the amount of perspiration. So effectively the sweat glands are activated. And using electrodermal activation, we can measure and monitor the activity of these sweat glands. So you can see if the conductance between these two channels that are attached to the finger, if that starts to increase, then it gives you an indicator that the sympathetic nervous system has been activated. And one of the things that activates the sympathetic nervous system is anxiety or a kind of fear response. So it might be the case that we use these measures to understand what emotional response somebody is experiencing. Similarly, we might use techniques such as heart rate variability. If you think that your heart beats in a consistent, regular, non-variable pattern, you're wrong. There's a lot of variability in the difference between the times of the heartbeats. And one of the things that we can do is record the exact timings of these. And we know that the more anxious or the more stressed that somebody gets, the more regular those heartbeats become. So again, we could use this to get an insight into, or certainly make an inference of what emotional state or level of stress somebody is likely to be experiencing whilst they're doing some sort of cognitive task. We might also want to look at hormonal measures of testosterone or cortisol. Cortisol, for example, is a good indicator of stress levels. So you can measure this using salivary swabs, and it gives you a very objective indicator of the amount of stress that somebody's recently undergone. We can also use imaging techniques. So again, there's going to be a lot of focus on this in next week's lecture. These include functional magnetic resonance imaging, or fMRI. We use electroencephalography, which is kind of surface scalp recordings. And similar to fMRI, there's a technique called positron emission tomography, or PET. And positron emission tomography and fMRI are both different techniques that can allow us to understand which regions of the brain are active while somebody is performing a task. So it's been really useful for understanding and localizing which regions of the brain are responsible for different sorts of cognitive tasks and cognitive processes. So some of the main strengths with using these psychophysiological approaches is one that they provide a sort of objective or hard evidence of different cognitive functions. So if we take the example of electrodermal activation, so you might have somebody come in and they're doing an experiment, and if you ask them at a self-report level, are you anxious or are you stressed right now? It might be the case that they don't understand or they don't have access to whether they are feeling stressed or aroused or nervous, whereas the electrodermal activation would give you a kind of objective indicator that their physiological system is responding to the stimuli. It's indicating that they are stressed or aroused in this particular situation. So it can give you decent insight into things that maybe people don't have accurate access to, or as some of the issues that we've already covered with introspection. It can give us insight into those functions or emotional states. Some of the difficulties or challenges with this, there are many. Two that I put down here is small sample sizes. So generally the challenges at a technical level and the amount of time it takes to set these up mean that you can't necessarily recruit large samples of people. Now the fact that it's difficult doesn't make a good enough excuse that people shouldn't collect big samples, but it's just the case that in a lot of the previous literature we generally find that people have run studies using sample sizes that are too small to make accurate judgments or accurate conclusions from. Okay, a final approach that's been really important in advancing knowledge in cognitive psychology, particularly from a historical perspective, is the use of individual case studies. So this can be an intensive study of either a single individual or a group of people that potentially possess some exceptional ability or some form of deficit. So one of the most famous case studies, we'll come to this in the lectures on memory, one of the most famous case studies in cognitive psychology is that of patient HM, or Henry Gustav Melason. So he had an operation to treat a particularly severe case of epilepsy. So it was a case of epilepsy where he was having fits and it wouldn't respond to conventional treatments, and they removed key parts of his medial temporal lobe. And it turned out that after having these parts of the brain removed, particularly a region called the hippocampus, patient HM was no longer able to form new memories. So he could retain a lot of the procedural memories that he already had, so a lot of the kind of motor skills and some of the existing semantic knowledge he had about the world around him, but he wasn't able to acquire any new skills or abilities, certainly nothing explicit. So we're going to cover HM and what his case can teach us about memory later. A little bit earlier before this, there was a patient who was referred to by the name of Tan, and he was called patient Tan because that was his only speech utterance he was capable of. So when he asked questions, his response would be tan, 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 tan. And he was a patient that was studied after his death by neuropsychologist Paul Broca, who identified that there were particular regions of the brain that were damaged in patient Tan. And these are now known as Broca's area. And we realized that this region is fundamentally involved in speech production. Now, one of the strengths of taking this approach is that it can provide causal insights into the localized function of different brain regions. So if you realize that a particular patient or a group of patients have injuries to a specific region, and then this consistently leads to an impairment in a specific cognitive process, for example, speech or processing movement, or some other sort of visual processing. If we find that damage to that region consistently impairs this, then we can be pretty confident that that region is causally responsible, centrally involved in the process that becomes impaired.